Hey, I just wanted to quickly plug that if you guys want any tutoring, some new slots have opened. If you look in the description, there's a link to my physics and maths tutor profile in which I do tutoring on there. Hello everybody, welcome back to the A-Level Cookbook. Today we're going to be doing the OCR A-Level Biology A, paper 3 from June 2022, so let's get going now. So the heart can be affected by a variety of disorders, some of which involve the immune system. So figure 1.1 shows the roles of 3 different types of antibodies, R, S, and T. So you've got antibody R here, we've got antibody S, what's it doing over here? You can see it's binding to a lot of these pathogens here. R is chilling what? It's on the receptor of it's on the receptor of a white blood cell and it's got a pathogen there. And then T has got harmful molecules released by the pathogen. This antibody's kind of got it like tagged onto there. So we need to figure out what the names of the 3 different types of antibodies are in figure 1.1. So if we look at R, you can see there's a pathogen here and it's tagged with an antibody and there's a receptor here. So in this case, you've got opsonization going on. It's labeled it, and the phagocyte's like, whatever white blood cell it is like, yo, I got to deal with this. So it's tagged it, so that's opsonin or opsonization. So when you have opsonins, what happens is you bind to a pathogen and you tag them, and then they bind to a receptor on a phagocyte, and phagocytes have those sort of complementary receptors to that, and then engulf and digest it. Immunoglobulin G and immunoglobulin M are the ones that have the strongest opsonin effects. So in S, you can see you've got an antibody and it's clumping together these pathogens here. So that means it's agglutinating. It's putting them together and sticking them together, clumping them. So agglutinins. With T, we've got harmful molecules released by the pathogen and this antibody is binding to them. Those are toxins, so it's acting as an antitoxin. A condition called rheumatic heart disease can occur when people's antibodies attack antigens on their own heart cells. What type of disease is this? So it's an immune response to your own body. So it's autoimmune. So now it says figure 1.2 shows 2 electrocardiogram traces, an ECG of a normal heart activity and an ECG of a person with a high type of heart disease. Describe how the ECG trace of the person with the heart disease is different to that of a normal heart. So you need to be able to recognize what, each of these waves mean. So that's a P wave. Sorry, I'll make this a bit clearer. So that's a P wave, and that's atrial depolarization. So the atrial contraction. This is your QRS complex, and that's where you have, so the little thing here is Q, that's an R, that's an S. That's where you're having ventricular systole or contraction, right? And during this, you have the atrial relaxation. You can't really see that on here because this overshadows that. And then here, this is your T wave. That's your ventricular repolarization. They're chilling, they're relaxing, they're going back to normal. So you can see there's a P, Q, R, S, T, nice gap, P, Q, R, S, T, and so forth. In this example, you can see a lot of things going on here. So the general rule of thumb is find the QRS and then look for the P's before it, because the QRS will always have a P before it. It's a P, QRS, and then a T's mishmashed in here, and then you've got P, P, or, PQRS and so forth. So we can see that there's not a clear, nice PQRS. In the other one, in this person who's got the heart disease thing, remember that the distance between each of these also tells you a bit about its rhythm as well. The QRSs, I can say off the bat, I should have really said that first. The QRS complexes are closer. And then as well as that, you can see that they're also differing, they're irregular between them. So there's irregular gaps or distant times between the QRS complexes. Here, like I was saying before, it's really hard to tell if there's any clear P waves. So there's no clear P waves. And what else can we say? And also, as well as that, there's no clear T waves. So actually, it's pretty not good. This person probably has atrial fibrillation, for anyone that is geeking and wants to know. So then gene therapy is a possible future treatment for heart disease. The AC6 gene encodes for one form of the adenylyl cyclase enzyme. Clinical trials have tested the effect of increasing the levels of AC6 gene in the heart cells, suggest how using gene therapy to increase the levels of the AC6 gene may improve heart function. So again, with this kind of stuff, what I like to do is I like to think, what's the end consequence? What are the facts I can establish from here? Because you probably have never heard of this gene, and they're not expecting you to. They're expecting you to think in your head and be like, what can I apply here? So what they've said is that if I increase AC6, for some reason that improves heart function. Right? Now, again, only on what the question's saying. If I increase AC6, what does that mean? It codes for one form of the adenylyl cyclase. I'm going to say adenylyl cyclase. So that means if we're doing more, if we've got more AC6 gene, we've got more adenylate cyclase, right? So cyclase. That's all we've got. That's all the questions told us. And they just told you that if you increase AC6, and that must mean you're increasing adenylyl cyclase, and so for some reason that improves heart function. So now we need to think, okay, what possible explanations do I know? It's a suggest. So we need to think, where do I know adenylyl cyclase? It's in your second messenger model. So that means we have more adenylyl cyclase. And what does that do? It converts ATP into cyclic AMP, sorry. So that means we've got more cyclic AMP, right? And you need to think, okay, well, why does that all matter? What's all this related to? This is all to do with adrenaline. So adrenaline works through the cyclic messenger model, which involves adrenaline cyclase. So adrenaline binds to your receptors on heart cells, and because now there's more CAMP than usual, what do you think might happen as a result? So obviously we're familiar with it doing a cascade of proteins and this and that, blah, 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 and eventually get glycogenolysis and so forth. But this is telling you that it's improving heart function. You need to think, how does that improve function? What's the function? So in this case, it must be that it's increasing heart contraction, because that's what makes a functional heart a good fat heart, one that beats fast and one that beats hard, right? As long as it can actually do both of them and has a balance between them. You don't want to go too fast. But anyway, that's the point. My point is you've got to look at the question, establish the facts, then fill the gaps. So that's a state one method for inserting the ACE6 gene into heart cells during gene therapy. So your options are a viral vector, or you could use a liposome. So I'm just going to write viral vector. Now it says the results from the gene therapy trials are published in peer-reviewed journals. Suggest why the results from gene therapy are published in journals. So it's just to be able to, first of all, share new knowledge. No point gatekeeping it, you know? But also, it allows validation as well of it, of the knowledge. Because what that means is you've now got all these other, nerds that are like, yo, I absolutely love the heart. Whoa, that's crazy. And they can look at your experiment and be like, wait, hold up, this is a terrible experiment. What the hell? Or they can talk about it and critique it and suggest better ways of doing it and take it and do better things. So that's why it's important. So now it says figure 2.1 shows a light micrograph of a blood smear. The cells labeled X and Y in figure 2 are two different types of white blood cell. Identify their types. So if you look at like the different white cells you've got options like, for example, neutrophils. So they've got a granular cytoplasm, multi-lobe nuclei, and so forth. So if we drew that as a diagram, neutrophils have sort of, it's like you've got your different lobes here, and you've got all these little granules filled in them. So if we look at X and Y, we can see Y has got those multi-lobed things. The granules are hard to see, so that must be a neutrophil. And your lymphocytes tend to have large nuclei that fill most of the cells. They're kind of chunky like that. So based on that, X has got to be either T or B lymphocytes, just lymphocytes. So now it says the blood cell label Z in figure 2.1 has a high concentration of hemoglobin. Outline two other ways in which it is adapted to its function. So you should immediately recognize, it's told you that it's got a high amount of hemoglobin. You should also be able to tell that that is a red blood cell. So there's multiple things you can say. So you can say that it's flat and biconcave. So what that means is if you look at its side on, it's got like a dip like that and a dip like that. It's exaggerated, but yeah. That means what? It increases its surface area to volume ratio. There's no nucleus. You need to say what that means. More space for hemoglobin. Because that's talking about the structure, that's how it helps its function. Anything else you could say? There's other things as well. I'm just going to put two, but you could also say that they're flexible, so they can squeeze through capillaries. You can also talk about the fact that they allowed chloride shifts because of the transport proteins in their cell surface membrane. They have a high concentration of carbonic anhydrase within them, so it allows you to transport CO2. They're quite small as well, so there's a small diffusion pathway from when you're getting oxygen in and out of them. So then it says the diameter of another blood cell represented by the line W in figure 1, the magnification was times 800, calculate the actual diameter. Because I'm on a screen, it's really hard to do, so I'll just describe what I would do. So the image size, I, equals actual times magnification, I am. So we know that it's times 100. So it'd be times, so we know that's 800. The image size is whatever you've measured. So you need to use a ruler and measure it. So I'm going to pretend that, I don't know, I'm making this up, that's like 1 centimeter. So 1 centimeter, and the actual size is something, and that would be in centimeters because the units have to be consistent. Once you've figured that out, you would do I over M, and that would give you the actual size in the same unit as that, so centimeters. Then you'd have to convert it to micrometers. So we did that. To get from centimeters to millimeters, you times it by 10. To get to from millimeters to micrometers, it's times another 1,000. And there you go. So now it says some white cells have a high concentration of lysosomes. State the role of lysosomes in it. All it does is it just hydrolyzes or breaks down or digests or whatever you want to say, pathogens. Next, it says a scientist calculated two values for the lysosomes in a white cell. So the mean volume of a lysosome was this. The mean number of H+ ions per lysosome was that. Use these values to calculate the mean concentration per lysosome in this white blood cell, and then give your answer to moles to decimeter cubed. So they've told you that the H+ ion number is 1.3 times 10 to the minus 21 moles, and they want your answer in moles per decimeter cubed. You can see that's in centimeters cubed. That's a volume. That's a volume. So you could probably figure it out by saying that's moles divided by sewing in decimeters cubed. But as well as that, if it's concentration per lysosome, it's going to be how many of these can you squeeze in a given volume. So the volume is going to be 6.5 times, so 6.5 times 10 to the minus 14 centimeters cubed, but they want it in decimeters cubed. The way I think of it is that 1 decimeter is 10 centimeters, right? So you're timesing that by 10. So if you got 1 decimeter squared, that's going to be, so you're going to times it by 10 squared, 100 centimeters. If you got 1 decimeter cubed, you're going to times it by 10 cubed, so 1,000 centimeters, squared and cubed. And so then this would be 6.5 times 10 to the minus 7, because of dividing it by 1,000 decimeters cubed. So then to work out the mole concentration of moles per decimeters cubed, it's 1.3 times 10 to the minus 21 over 6.5 times 10 to the minus 7, which is, wait, why have I said 7? 17, sorry, oh my god. So that gives you 2 times 10 to the minus 5. There you go. So now it says the formula used to calculate the pH is that negative log to the base 10, where that's the concentration in H bar, where H plus is the concentration in moles per decimeters cubed. Calculate that. So all you got to do is you got to stick your answer into negative log to the base 10 of 2 times 10 to the minus 5. And if we do that, you get 4.7. So now it says the scientists stain the lysosomes in a sample of living white blood cells, and the table shows some of the properties of 5 stains A to E. Select the most appropriate stain for the scientist to use based on your answer from part 3. So in our part 3, we have said that its pH is 4.7. So it's suitable to stain alkaline components? Nah, because that's less than 7. Acidic, probably looking good. Neutral? Nah. Alkaline components? Nah. Acidic components can be used to stain fixed sections of tissue. Well, these are living cells, and we're not using a section of tissue because that'd be dead, and it'd also probably not be doing very much, and we're also using lysosomes as well in a sample of blood from somewhere. So it's probably not best to do that either because it's not fixed. It's a sample of blood that's been smeared across a thing. So it'd be. So it says differential staining can be used to distinguish between bacteria with thick cell walls and thin cell walls. 4 substances which are differentially used can be crystal violet, which stains bacteria purple, safranin, which makes bacteria pink but not visible in the presence of crystal violet, alcohol, which removes fixed stains within thin cell walls, and then iodine, which fixes crystal violet to bacterial cell walls. Suggest a practical procedure that would allow you to distinguish between thin and thick cell walls. So we're distinguishing thin versus thick, right? So it's told you that bacterial will be stained purple if you use crystal violet. So the good start is probably that, because now you've got purple and purple. Now, safranin can differentiate between these two. Sorry, not differentiate. Sorry, safranin, no. Safranin can stain them pink, but it doesn't work if you've got crystal violet there. So what we could try to do is look at what other options we have. So if we have alcohol, it will remove fixed stains from thin cell walls. So alcohol can help distinguish between these. Issue is that this isn't fixed. If you use iodine, it will fix it. So the next step would be that you use iodine. So again, you've got your crystal violet and your crystal violet. But then if you add alcohol, it says it removes the fixed stains from thin cell walls. So we can add alcohol, and now it's not got that color anymore. That's purple. But then we also want to make sure there's still bacteria there. So then you can add safranin, and that'll make it go pink while the other one's purple, and now you can distinguish the two. So you'd say like that. So first it's crystal violet, then iodine solution, then alcohol, and then safranin. So figure 2.2 shows stained tissues that includes different blood vessels, L and M. Where are we? So there and there. And a substance labeled N. State which one it is. Is it an artery or vein? And give 2 pieces of evidence. If we look at L, you can see it's quite a thick wall, and the lumen is pretty small. Compared to where are we? Sorry, M, it's pretty wide and thin. Remember that this is a cross section, so it's not going to always look like a perfect, nice little thing. It might be that this is a bit of an angled bit, or it's going like that way. So it's actually cut sideways like that. So just remember, that doesn't mean that it can't possibly be a vessel, because it's not a nice tube. So based on that alone, it's an artery because it's got a small lumen or diameter, and it's also got thick layer of smooth muscle. So now it says state the substance labeled N. So we've got this N here chilling. What's that? It's a smooth muscle, and then it's got this around it, and it's all sort of all over the place. So it's something that's found around this smooth muscle, and it's providing structural support, because that's why else would it be so nicely arranged around that? It is collagen, because collagen does exactly that. So generally speaking, vessels have varying amounts of elastic fibers. It's made of elastin, and that can stretch and recoil, and that can provide vessel walls with stability. Smooth muscle, which contracts or relaxes, not expands. All muscles in the world can only contract or relax. What that does is that can change the size of the lumen and helps maintain pressure, and collagen is around it to help provide structural support, so it keeps that nice and open and stuff like that as well, and helps it not get too big, too small, and kind of, yeah. A student wrote a method for taking a cutting to clone a plant. Select the stem with many flowers and leaves, make a slanting cleft in the stem below some leaves, dip the cut in stem in rooting powder, and then plant the cutting in watered compost. Describe and explain how this could be better. So when it comes to cloning and taking cuttings in this sense, there's a couple things you can do to make things better. So there's a couple rules. Number one, you should try to use a non-flowering stem, and you need to explain why. You might need to think, why doesn't it make flower leaves and stuff like that? It's because it will reduce water loss, and it will help encourage root growth. And then again, for a similar sense, you want to remove leaves as well. Why? For the same reason. Then what else do we want to do? All they've done is you've got to work with what they've said in the question. They've literally just done this, chopped it, taken it, this and that, blah, blah, blah. Have we talked about anything to do with like sterilizing, what they're actually doing? So we need to make sure that we're using an aseptic technique to avoid contamination. So you can't just say do it, you need to say why. Contamination. And then we need to think, okay, well, planting it in watered compost, and they're looking to see if they want to clone a plant. Low-key, like you're just putting it in there, like, how do you know it's going to grow? So you could use, like, for example, a greenhouse, a propagator, or you could describe it, so you could grow it in controlled temperatures, things like that. And as well as that, you don't want to overwater the compost, because they've told you it's watered compost. If you overwater it, then you're not going to have as much oxygen in there. It's going to be waterlogged and it can't respire. So you got to look at each step and critique it and think, what can I do better? What's wrong with this round? What could screw up the results? Or whatever it is they're doing. So the student investigated the effects of auxin concentration on the growth of shoots. The student applied different concentrations of auxin to the apical shoot and the lateral shoots, and they measured the percentage of growth stimulation and inhibition compared to normal. So normal growth was 0, which is right here. So if we look at this, we can see that the lateral shoots have a positive stimulation growth. There's more, there's more growth going on, and then it goes down. Fine. Then if we look at the apical shoot, it starts, the stimulation of growth starts a little later, and it goes up, and then shoots down. Don't know why, haven't thought about why, I'm literally just making sense of the results. So now it says, use the graph to estimate the auxin concentration at which the inhibition of lateral shoots is 100%. So inhibition of lateral shoots, 100%. So we're looking at the 100% and we're looking for lateral shoots. So what it means, we have to estimate this by looking at its trajectories. It's going up and then down. And it looks like it seems to be shooting down like that. So what you could do is draw like a nice little line of best fit through there and estimate it. So minus 100, 10 to the minus 5. The student identified a possible anomaly in their results. The data point for the apical shoot receiving 10 to the minus 6 suggests what they could do to determine whether this was an anomaly. So if we look at 10 to the minus 6, where are we? That's the minus 6. That's my glasses are there. So how can they determine whether they're anomalies? So imagine you flip a coin and you get all 10s and you're like, that's a bit sus. Why have I got all 10s? You'd just do it again, wouldn't you? So you would repeat measurements for that concentration. Because then if you kept doing it over and over again, you got the same thing, then it probably isn't an anomaly anymore. There's something wrong with your method. So in the same sense, if I flip a coin 10 times again and now I've not got all 10s, it probably was just an anomaly. Using the graph, describe the conclusions that could be drawn about the role of different auxin concentrations in the control of apical dominance. So what they're trying to say is, what effect does auxin concentrations have on apical dominance control? It's saying to use the graph. So don't just dive in being like, oh, well, this happens, and this happens, and this happens from the notes, You've got to look at this and figure out what's going on. So apical dominance is where you've got high concentrations of auxins in the growth of lateral shoots. And what that does is those high concentrations suppress the lateral growth of lateral shoots. So we need to find the point at which we're seeing that happen. If you've got the suppressed growth of lateral shoots, it means your apical ones are going to be growing more. We need to look at this data they're So if we look at our apical shoots, you can see that it started to climb up here and then shoots up and hits the peak and then it starts dropping again. So from about that 10 to the minus 9 point, you must have apical dominance. So at concentrations higher than like 10 to the minus 9 to what? Like 10 to the minus 4 because it starts to drop? Apical dominance is present or is established or whatever you want to say. Right? But then that only explains why it shoots up. Why is it all of a sudden going down? You need to look at this data and think of a possible explanation. But at very high concentrations after 10 to the minus 4, the apical stimulation of growth lowers. So therefore, you could conclude from that very high concentrations inhibit apical shoot growth. It's asking you to use that data and draw conclusions. No other thing you can say from that, because we're looking only at this data here. And remember that, like I said before, with lateral shoot, when you've got that apical dominance, the lateral shoots are suppressed. So we need to look at what's going on here. So if we look at the lower, the lateral shoots, if you see at these lower concentrations, it's starting to get positive growth here, stimulation of growth. So at lower concentrations, lat shoots, growth is stimulated. I'm being very lazy with how I've done that. So it's like what, minus 10 to the minus 11 to what, like, it's still positive and then it starts to drop down to the negatives. It's still stimulated, remember, if it's positive. So that's what, 2, 10 to the minus 7. And then, well, we need to explain what we can conclude from here onwards. So it must be that above, like what, 10 to the minus 7, it must be that lateral, shoot growth is inhibited. So now it says another student plans to investigate the effect of gibberellin concentration on the rate of stem elongation in the pea plant. Suggest appropriate units for the dependent variable in this investigation. So we're looking at the rate of stem elongation, how long it's becoming, and it's a rate. So we're measuring it with respect to time. So you could do something like, I don't know, millimeters per hour. So the student has access to standard laboratory equipment and planting materials. Outline a method that the student can use to investigate the effect of gibberellin concentration and stem elongation on this. In your answer, you should include details of an appropriate statistical test for this investigation. So you need to figure out what the objective is. The objective is just you've got pea plant plus gibberellin, maybe makes it has elongation. That's the whole thing, the facts that you can establish from this question. Now you need to think of how you're going to do this as an experiment. So what we're doing again is we're looking at pea plant plus gibberellin elongation, and we're looking at if concentration changes the rate of it. So let's change that up a bit. So what we need to do is you get multiple pea plants. You can put a sample size in, high sample size, and you put them into groups. So what you can do is you give each group, apply each group different concentrations of gibberellin plus a control with no gibberellin. The reason is because we want to make sure that if there's any differences in these growths, it's because of those concentrations of gibberellin changing and nothing else. Because if your control starts having problems with it, then there's something wrong with your experiment that needs fixing. And you can talk about how you do that, so you could put it in the soil or put it on the roots and so forth or whatever. Fine, right? I'm just being very brief here. In the real thing, you can obviously write it all out. I'm just crash coursing it just for the sake of time because I don't want this video to be like 8 years long. Now you need to think about the variables. So with your variables, you can talk about the controls. So for example, you need to make sure you're using the same plants, the same seedlings, the same water, the same light temperature. Literally everything's got to be kept the same except your gibberellin concentration. And then as well as that, you need to figure out a way of how you're going to measure this. So what you would do is you could set them for a specific amount of time. So you could measure the stem length every 5 minutes, 10 minutes, and so forth, 6 minutes and whatever, right? Not 6, sorry, 5 or 10 minutes, and set an endpoint. So you'd be like, all right, after 2 hours, we're done. And then what you do is you put a table and put the length, the time, the rate, and so forth, right? And you could probably plot a graph. So you'd have a table of the concentration, you'd have the initial length, final length, time, work out the difference, and so forth, right? And then stats tests. So we're looking to see if concentration's affecting the elongation of rate. We're looking to see if the concentration of gibberellin affects the rate, right? So if we think about it, we're probably going to have the rate of the elongation here and the concentration, and maybe it goes up, maybe it goes down or whatever. So we're checking to see if there's any relation between this and this. As one goes up, the other one changes. So that's a correlation. So a statistical test you could do is a Spearman's rank correlation to see if any correlation coefficient, to see if the correlation between the rate and the concentration is statistically significant and not due to chance. And then other things you could do like a bar chart with means and stuff as well, and you can work out their standard deviations. So for example, if you had different groups, you had this concentration and then this, and then you work out the standard deviation bars and so forth. If they don't overlap, then that's different to that. The mean is different. But I think that's probably a reasonable one. And there you go. So it's just, again, identify the objective and then think what you would do. I'd be like, well, I'm going to change the concentrations because I want to see if that's affecting. So now it says a friend of the student had an apple tree in their garden. The friend asked if the student if there was a way to ripen the fruit more quickly so it could be ready to eat. The student said you should spray this with ethene. Evaluate whether acting on this advice would produce fruit that was ready to eat within a few days. So let's think about what's going on. So, well, ethene does promote ripening of fruits. That is correct. Again, we're looking at evaluating, so we're saying what's good and what's not so good, right? However, it's important to appreciate that the ripening of fruit is where you sort of get like this peak of ethene here, and that's what starts to do that, and you get loads of chemical reactions. What if they've already hit that peak? and you'll be past it, then it's probably not going to start making it ripe. So you need to know, the problem is, unfortunately, is we need to know the concentration of ethene, currently, and the stage of development, right? And additionally, where's the ethene actually acting? It's all within, the inside of it. Ethene is a gas, and it wouldn't work, it's not effective on the outside of the plant, or the apple in this case. And as well as that, it promotes fruit dropping as well in apples. So it promotes fruit dropping, and well, that's not great because they want it. What if they miss it and it's like damaged? I think that's a bit of a weird point to make, but there's no harm in doing it, right? So those are the things I would possibly say, to be honest. Is again, you got to appreciate that like that ethene is because of a peak, and then you get those chemical reactions that make something ripe. The sea sponge and the zebra shark are both animals. This sea sponge does not have an internal circulatory system. Instead, it filters food and oxygen from the surrounding water, as shown in this figure. So we've got the atrium cavity here. So the cavity is the atrium. We've got water flowing into the atrium through these tiny holes called ostia, and then water flows out of the atrium. And to suggest why it does not need a circulatory system, well, the reason we have a circulatory system is that because if simple cells can just diffuse things in and out if they need, because they're just one cell. Whereas when you have a whole, organism like us, it's got to get from the outside all the way to the inside and right into the core of us, and that's a very long diffusion distance. And the other thing is that with diffusion, as the gradient, gets less and less steep, it gets less and less quick. A high concentration gradient from high to low means that diffusion is very fast. But if you start to as it starts to level out, it gets slower and slower and slower. And you're getting to like really, really shallow concentration gradients. It's like barely anything going on. So we have that system in place, like the circulatory system, to make sure things are constantly moving, so they get around wherever they need to be, but also keeps concentration gradients nice and steep. Now, suggest why this doesn't. So they've told you it doesn't have a circulatory system, and instead, you can see it's filtering food and water from the surrounding areas instead. So you need to think, the only reasons that really could be the case is the opposite as to why we have a circulatory system for the reasons I just described. So you could say, for example, that it can get obtain nutrients and substances from diffusion due to a short diffusion pathway. Or you could say, for example, it's got a large volume to area to surface area to volume ratio, because you can see that's quite thin, and that's all it's got to get through. Now, the concept of surface area to volume ratio is quite something that I've seen people struggle on. They just kind of say it and not realize what they mean. Imagine I have a cube here, and they have the same area, and then I have another cube here as well, but it's got less volume. That has got a pretty big volume, and its surface area is this side times this side times this side, and then so forth. That means that diffusion can only come in through all the points on the outside like that. It can only enter through here, and it's got to get to its core in the middle like that. Whereas here, if the core is in the middle there, it can get through all these points and so forth, but it only has to go like, this thickness to get to the middle. So that's why high surface area to volume ratio is quite a good thing. And it makes sense because if you have like a piece of ice, for example, if you pick up a chunk of ice, it takes a couple seconds to melt in your hands. Whereas if you have like a really, really thin sheet, it melts really fast. That's because that thin sheet has a very high surface area to volume ratio. So now it says, a diagram of the circulatory system is shown in figure 2 of this thing, and you've got body tissues, veins, heart, the gills, and so forth. It says, describe the circulatory system that it has. So the two types is that you've got an open circulatory system, where you've got very small numbers of vessels, and what they do is it's pumped straight to the heart into the body cavity of an animal, and then this stuff starts to get into direct contact with the tissues and the cells. So that's typically things like your insects and stuff. Whereas in a circulatory system that is closed, everything is kept in blood vessels and you do not get direct contact with cells. Now here, we can see that you've got this sort of circulatory system going on here. And if I draw it diagrammatically for an open circulatory system, imagine that's your heart. Those are like your blood vessels, and it all eventually just comes back like that to the heart. Whereas here, in a closed system, if that was your heart, it would go to all the things, whatever, blah, 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 but then it would all come back in and then come straight like back into it in a circulatory system, like in tubes and pipes, in arteries and veins. So in this circumstance, we can see that this is a constant, it's kept within a circulation of its own, and it's within tubes, and it's a fixed system, so it's a closed system, right? So number one, it's closed. And then we need to think, is it a single or double circulatory system? So a double circulatory system is where in humans we have a loop that goes to our heart and a loop that goes to the rest of the body. So a loop that goes from our heart to our lungs and then a loop from the heart to the rest of the body. Here it is just one singular loop. There's no heart. So it's a single closed circulatory system. So now it says both of these reproduce sexually, but under particular conditions they can reproduce asexually. In asexual reproduction of this, it clumps from diploid cells detached from the body of the sponge, and then they reattach to a surface, and grow into new adult sponges. In asexual reproduction in the other one, meiosis occurs in a female. Two of the haploid cells produced by meiosis form a diploid cell, and then the diploid cell makes a new shark. A student stated that when they produce asexually, both animals produce clones of themselves. Evaluate this statement. So if we look at what they both do, this one, this one, what it does is you've got like diploid cells that come along and they detach and then they become new sponges. So the way that they must be doing that is mitosis. Whereas here, we've got meiosis, which makes haploid cells, and then they fuse, and you get a new shark. I wouldn't say both of them produce clones, to be honest, because if we look here, if we look at the sponges, like I just said, the sponges, they're going to produce genetically identical clones as their offspring undergo mitosis. Things that divide and replicate like that, they're going to be identical clones. Whereas in the shark, the offspring may not be identical genetically. And the reason is because if we have a look, they're undergoing meiosis. So it's due to meiosis. Now, in meiosis, we have independent segregation of homologous chromosomes and crossing over, which introduces genetic variation. That's what gives us those differences. I'm going to quickly describe meiosis as well, just real quick. So we have like a cell, and what it will do is, so if imagine that's your chromosome, and that's another separate chromosome there, in a cell. In meiosis, what happens is it will make a copy of either one of them like that, right? So these are still chromosomes. There's just two chromatids that make them up now. And then what happens is, imagine that's your maternal one, sorry, that's your paternal one. That's your maternal one, that's your paternal one. What will happen is that, okay, so quickly just meiosis, if you have like a normal diploid cell that's chilling, you have a maternal chromosome, you have a paternal chromosome, and then you have another maternal chromosome and another paternal chromosome. Like, well, we have 23 pairs, but I'm just pretending this organism has 4, right? So what will happen is, in meiosis, first of all, we need to make a copy of each one of these. So what it will do is it will do this for either, and those are your two sister chromatids, but you can see that's still maternal, maternal, paternal, paternal, maternal, maternal, paternal, paternal. Those are your two sister chromatids, and they're bound together by a centromere. That's what happens in the state first prior to meiosis. In the first division of meiosis, you get homologous pairs separating. So homologous means the things that correspond with each other. So if those are chromosome 7s, imagine that's chromosome 7, that's the chromosome 7, those are a homologous pair, as in the paternal 7s and the maternal 7s, of which we have copies. So what the first division does is it will separate the maternal and paternal copies, like so. So paternal, 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 maternal, 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 right? And what also happens in that first division is you've got those crossing over. Otherwise, you just have the identical things passed over. So what happens is these will if we zoom in on them, they'll cross. So crossing. So if we zoom in, maternal, and then paternal here, there, they'll cross over and exchange bits and bobs like alleles and stuff, and then they make these new things. So that's what happens there, and that's one way we get genetic variation. In meiosis 2, what happens is these chromatids, those double-armed chromatids, now separate. So you get 1, 2, 3, 4 with maybe it could be a maternal paternal, and then like a maternal paternal, like, sorry, not maternal paternal, maternal maternal, maternal maternal, paternal paternal, paternal paternal, right? But obviously we've exchanged alleles across the two of them, so they're not identical to the original ones there. And that's what happens in meiosis too. That's what gives you that sort of haploid cell, because can you see there's only maternals and only paternals there. Whereas in mitosis, In mitosis, the homologous pairs won't separate. You're just copying everything in there and then just making two diploid cells out like that. So that's what makes it different. Because here we're still diploid, maternal, paternal. Here we're still diploid, maternal, paternal. Here we are haploid because we're now maternal, maternal. We would have 2n, and then we'd have 2 times 2n, 2n meaning maternal, paternal, two lots of them because we've just doubled them. This would be 2 times n because we've only got maternal and paternal. We've just got doubles of each, and then these would all be n in one. So it says humans can produce artificial clones of animals. Describe 2 methods for doing this. So the 2 methods that we can do is we can do something called somatic cell nuclear transfer, which is where you basically just take out a nucleus from a somatic egg cell, you then stick it into an egg, you do something called electrofusion, and then you put it in surrogate and let it grow. Or you can also do something called artificial twinning as well. So artificial twinning is where you sort of in natural twinning, what happens is if you have an early embryo, it will split, and then you get two halves of a divided embryo, and they become two separate fetuses. Artificial twinning is exactly that same thing, forcing that to happen and making it happen. So we'll describe each one then. So what happens is one of them we can do artificial twinning. In artificial twinning, what's happening is that we basically so you need to describe the steps of actually inducing that. So again, like I said before, when you have when an egg is fertilized, it becomes a totipotent cell. It can make literally anything in the universe, and then a couple divisions after, it can't do that anymore. So in natural twinning, that early stage, it splits into two. So first of all, we need to generate an embryo. So what we do is you'd pick like an organism or male or something like that with desired characteristics, and you take sperm out of them. So you take sperm out of male animal with desirable characteristics. Right? And then what we need to do is we're generating the whole embryo, we're doing that. So what you need to do is you can do in IVF. So that's basically you're artificially fertilizing an egg. Right? And then what we do is we then split them. So how we do that. So then what this will do is it'll start to divide into an early embryo, and it's got nice little bits and bobs in and stuff. We want to split it before it's too late. So then what we do is we split the embryo is split, and then it's almost like you've got separate now different embryos chilling, and then they can start to continue to divide and stuff. So then what we need to do is actually make them grow. So we implant these in a surrogate, and that produces clone offspring. And that's how twinning happens when you've got, humans, for example. You've got like that fertilization, it starts to divide as it normally would, but then it kind of splits for some reason. Not true. Yeah. And then here you go. You've got two separate things, which will then divide into two clumps, and now you've got two separate organisms growing, and that's what gives you those twins. And those are identical twins. You can have fraternal twins as well, where you have two eggs that fertilize separately. That's different because they're not identical genetically. These are genetically identical because every single thing that they originally came from was the same fertilized egg. So then the other option is somatic cell nuclear transfer. It's in the name. Somatic means body, like just generally body cells, somatic cells, soma, body. Cell nuclear transfer. You can figure out what it means there. It means transferring the nucleus of a somatic cell. So what you would do is you want to collect a nucleus. So remove a nucleus from a somatic cell. And then what you need to do is we got to stick it in an egg. So you enucleate an egg. That means basically remove an egg. It's a nucleus, sorry. Enucleate an egg. And then insert the somatic cell nucleus. And then in order for it to actually make it do its job and like sort of, in order for it to actually take up the egg and then start dividing in the same sense as like a normal fertilization egg thing going on, we need to give it an electric shock. So an electric shock is given to take up the egg and start dividing. Because in your somatic cells, you're already diploid. So it's almost, in that circumstance, you don't have to put a fertilized egg You don't have to fertilize with an egg and a sperm because egg and sperm are both haploid and they come together to get a diploid. But we're just taking a nucleus that's already diploid and ready to go. So then so the electric shock is given to take up the egg and start dividing, and then that process is called electrofusion. Then what happens is, again, in the same sense, we actually need to provide it with some way of growing. So you transfer the embryo into a surrogate, and that'll give you identical clones to the original surrogate. Sorry, not the original surrogate, the original somatic cell. So that's that, but then so these clones offsprings, I should have really said, they're clones of each other. So what that means is like that doesn't if those are clones of each other, that means that they're not clones of the parent. They're just clones of each other. So for example, it's like saying your mom and dad have had you as twins. That means you guys are genetically identical to each other, but not to your parents. You're different from your parents if you're twins, and you're identical twins. Whereas somatic cell nuclear transfer is literally taking their cell, putting it in a thing, and making it grow again. So you will become genetically identical to the parent. That's different. The only thing that we've ever done that with successfully, I think, to my knowledge, is Dolly the sheep, but we haven't done that in humans, and I think I can kind of see why that might be a bit odd. So now it says DNA must be extracted from cells before it can be analyzed. Sentences describe how DNA is extracted from a sample of tissue. Complete the sentences using the most appropriate words or phrases. So what we do is we've got detergent is used to break down something. So we keep all that DNA in the nucleus. We need to grab it out like that. So it's going to be the nuclear membranes. So proteins such as histone surrounding DNA can be hydrolyzed with the addition of proteins hydrolyzing them, proteases or protease. And then the DNA is precipitated from solution by adding, and it's ethanol. That's how they do it. So now it says DNA analysis can be used to assess genetic biodiversity within populations. A scientist assessed genetic biodiversity in 4 populations A to D of yellowhorn, which is a small tree. They used 2 measures of genetic biodiversity, so the percentage of polymorphic gene loci and then observed heterozygosity. So it's the proportion of heterozygous loci in a population, and they analyzed 23 gene loci in each individual tree they sampled, and the results are shown in this table below. Another scientist stated that these results may not allow an accurate assessment of genetic biodiversity in these 4 populations. Give 2 pieces of evidence that support this. So what are they trying to do again? They're basically doing DNA analysis to test biodiversity in a population, right? And this person's saying that these results might not allow for that. So if we have a quick look, we've got population A, they've sampled 6 trees, and you can see that there's 86.96% of polymorphic loci and observed heterozygosity, 0.68, whatever, fine. Here they've done 16, and they're seeing that 100% of them have polymorphic loci, sorry. So polymorphic genes is where you have more than one allele. So for example, when we have like our blood types, we have blood type A, B, and O. That's polymorphism there. There's 3 different types. It's a polymorphic gene. So in this sense, when it's talking about polymorphic loci, it's talking about the percentage of them having different alleles. So 16 there, we've got 6 there, and it's 91.3%, and then 6 there, that is 100%. So what kind of problems do we have? Well, they're trying to, again, look at genetic diversity within populations. They've done 23 gene loci in each tree. Well, I mean, first of all, they've only tested 23 different loci, as in 23 different genes. There's probably more than 23 genes in, the entirety of a tree. So only 23 loci are tested. As well as that, I mean, 6 trees. It's a pretty small sample size as well. So it's just asking you to critique what might be going on, and that's why it might not get accurate. Because, for example, if I have a room full of 10 people, I give them chocolate, and all 10 people shit themselves, that's 100% people shitting themselves after they eat chocolate. If I then go and tell the world, hey guys, if you eat chocolate, then you're going to shit yourself, and that's a guaranteed thing, you guys don't think that happens. And why does that not happen? Because the sample size is terribly small. What if by pure luck or by chance, that's what I got? So, yeah. And maybe if I did a statistical test, it would show me my p-value is greater than 5 for that observation versus expected result. And, you can see it come into play there. So that's how the sample size is. So it says the Hardy-Weinberg principle can be used to calculate allele and genotype frequencies in the populations. The common morning glory plant, the Ipomoea alporia has a range of flower colors. Two flower colors, purple and pink, are determined by a single gene. So big F is dominant, and that's purple. And then little f is pink. It's always good to just scribble that down so you really make sense of what's going on here. Now, it says here, a field contains 600 alporia plants. So the total is 600. And it's told you 150 of these are pink. So the genotype for that has to be little f, because pink is recessive. So the other 450 are purple. So they're purple. Sorry, whoops. So it could either be big F, little f, or it could be big F, big F. We don't know. It's either one of the two. The question says, using the Hardy-Weinberg principle, calculate the number of plants that had a homozygous dominant genotype. And they've told you to use these equations. So in the Hardy-Weinberg principle, what you're doing is you're working with allele frequencies, which is how often an allele appears in a population, not the raw numbers. And the reason they add up to 1 is because 1 as a decimal is 100%, right? So these should, like these different genotypes add up to 100%. So that's just a singular allele. So it could either be a dominant or a recessive, whichever one. And they should add up to 1. Because in a population, all the alleles that you see should add up to 1. And if it changes between generations, as in, like, for example, this goes higher or lower, it means either new alleles are coming in or old alleles are going out. And the reasons for that could be, for example, migration, births, deaths, mutations, natural selection, all kinds of stuff like that. In theory, if you have one generation of geezers, the next generation of geezers should have the exact same proportion of alleles. So, for example, the number of, like, I don't know, tall ostriches and featherless ostriches is probably going to stay the same. If it's not, then those factors are coming into play. So if we're using the Hardy-Weinberg principle, this only works if we use, again, like I just said, proportions. So we have 150 of these that are pink. So to work out the proportion of them and the frequency, you do 150 over 600. And if we simplify that, you get a quarter. And then 450 over 600 is 3 quarters. So 450, which 3 quarters is 0.75, and that's 0.25, right? So then what we do is we need to try and figure out what these we have. So remember, let's understand what's going on here. So I'm going to call that one my P, that one's going to be Q, right? So if 450 are pink FF, that means that QQ equals 0.25, right? It's Q and a Q, so it's Q squared. So that means Q would be the square root of 0.25, which is 0.5, right? Now that we have that, in theory, it wouldn't work if it didn't, if this, this equation wouldn't work if this wasn't the case. P plus Q is 1. So that must mean that P would equal 1 minus Q, which is going to be 1 minus 0.5, which is 0.5. Okay? Good start, right? So, now we know what P is as well as Q. They're both 0.5. But we need to figure out the ones that are homozygous dominant. So if we look at P squared, that is the same as saying FF plus, and then 2PQ would be big F, little f, and little f, big F. Can you see those are the exact same? That's where that 2 comes from. And then the Q squared is FF squared, little f squared. So again, if this equation holds, and it should do, you can just do you can just take away the 2PQ and the Q squared, or you can just square the P that we've now just worked out. So if you do P squared, that's going to equal 0.5 times 0.5 squared, which is 0.25. And that's the proportion of them, remember? But they want us to find the number of plants with that genotype. So therefore the number of plants would be 600 times that 0.25, which gives you 150 of them. You could work out P squared, you could work out 2PQ plus Q squared and minus it from 1. But again, like if we're using the Hardy-Weinberg principle, you can just do P squared, or you could work 2 times P times Q out. So now it says hemoglobin is an important protein in many animals, including humans. Sickle cell disease is caused by the abnormal production of abnormal hemoglobin, A treatment of sickle cell disease is based on a technique called CRISPR gene editing, and it allows SCD to start producing fetal hemoglobin, and it's done by these steps. So bone marrow stem cells are removed from people with SCD. An enzyme called Cas9 is added to them. Cas9 deletes the bases from the BCL11A gene, and this BCL11A gene usually switches off the fetal hemoglobin gene in adults. So if we're cutting that out, it means that fetal hemoglobin gene is now on. The gene-edited stem cells are then placed back into the patient, and the patient can now produce fetal hemoglobin. The CRISPR gene editing method is different from traditional genetic engineering, which uses restriction endonucleases or restriction enzymes, and plasmids. Describe the similarities and other differences between CRISPR gene editing and traditional genetic engineering material. If we're comparing them side by side, CRISPR, if we just have a quick look again, it uses Cas9 and that deletes bases. It just removes them, right? If we look at our normal sort of traditional methods, restriction endonucleases do also cut DNA. They break them, remember, from at sites where they can recognize them. So this works by cutting DNA at palindromes. So there's a similarity there. Is that the one similarity already, if I scribble that down here, is that both of them cut DNA. They both remove them, right? And as a result, they're going to both change the base sequence, right? Because if you're cutting this and then we're doing all the other stuff with the plasmid and stuff, you're eventually going to change the base sequence. If they both change the base sequence, because we're talking about hemoglobin, which is a protein, all proteins are made from amino acids, and the sequence of amino acids is determined by their base sequence. So if you're changing the base sequences here, you're going to start producing different proteins as a result. Those are their similarities. However, if we look at our differences, this is literally just deleting things, whereas here we cut them, and then what we do is if we're going to use plasmids, we insert these into plasmids. So here is literally just chopping things out, whereas here they're inserting it. So the difference is that Cas9 only deletes bases. Our traditional methods insert new ones. So you can do a lot of this just by looking at what the question is, as long as you have a vague idea of how these work. As well as that, as a result, the Cas9 doesn't need or doesn't use a vector. You're just taking the cells and you're directly editing them by taking the genes out. Whereas in traditional methods, you've got to use the vector, because what you're doing is you put them in plasmids, you put them in a bacterium, you let them grow, and so forth. As well as that, you wouldn't use DNA ligase in this either, because it's not sticking things together. And those are the points that I would make. In the Mark scheme, it says, as well as that, traditional genetic engineering is illegal in humans as well. That's nothing I guess you could say, but those other things enough is enough. I wasn't really thinking that. Hemoglobin plays a crucial role in transporting oxygen in animals. Several ions also have roles in oxygen transport. 3 ions are listed in the table below. Place ticks in the correct boxes to indicate which properties and features are true for each ion. So just as a quick reminder, let's just have a quick crash course on what happens with your hemoglobin and all that kind of stuff, because you can see it says hydrogen carbonate and so forth. So when you have respiring tissue, aerobically respiring tissue, I should say, what it does is it will release CO2. Now, we don't have CO2 bubbles floating around in our body in the same way like a Coke can does. So what it will do is this CO2 dissolves in water, and that will produce carbonic acid. Carbonic acid. And this is catalyzed by the enzyme called carbonic anhydrase. Right? Now the rest of so that's like about 90% of your carbon dioxide. The rest, around that 10%, goes directly to the lungs via hemoglobin. Now, the reason we do this is just because it forces it to get to the lungs in a better way. If we just had bubbles of CO2 floating in our blood, you'd have to wait for it to eventually just hit the lungs, and that's just a bit pointless. So that's why this happens. So if that goes directly via the hemoglobin to the lungs, let's talk about the journey of what happens with that carbonic acid. So this carbonic acid, it's H2CO3. So what it will do is it will split into protons, H plus, and what we call hydrogen carbonate ions, HCO3 minus, right? And what happens is that if we have an increase in H pluses, that actually decreases the pH. So we want to try and do something about that, because we don't like having too much, acid in our blood. So what happens is that oxyhemoglobin, I'm just going to write oxH because I'm lazy, it unloads its oxygen, and then it sort of it takes up the H plus, and that forms hemoglobinic acid. And that again, this also stops the cell becoming more acid, as well as also the blood more acidic, because again, more H plus means more acid. What happens is we need to also sort out what goes on with this HCO3. So the HCO3 minus is diffused out of an RBC, red blood cell, into the plasma. And when you do that, we need to compensate. You can't just you can't shove out something with a charge and then not have another thing come back in. That doesn't balance the charges out. So what happens is to compensate, Cl minus moves into red blood cells, and that's what maintains the charge. It keeps everything nice and balanced, and that's called your chloride shift. So that's that happening. We're getting the HCO3 in the blood plasma traveling to the lungs, and some CO2 as well. So when the blood gets to the lungs, remember the lungs breathe. Well, when we in our lungs we breathe out CO2, so we need to get it back into CO2 form. So what happens is that some H2CO3 and the sorry, not the H2CO3, some of that hydrogen carbonate HCO3 minus and H plus combine, and they give you CO2 and H2O, the water. And that CO2 can now be, diffused into the alveoli and breathed out. And that the other carbon dioxide that's directly in the hemoglobin also does that. So that's a quick crash course. As a reminder, you respire, you release CO2, it dissolves in water to form carbonic acid, which is catalyzed by carbonic anhydrase. The rest of it directly binds to hemoglobin and goes straight to the lungs. Of that carbonic anhydrase, it splits into H plus and HCO3 minus. The H plus makes oxygenated hemoglobin unload its O2 and then take the H plus up to stop it from becoming more acidic because there's more H plus. And then that forms something called hemoglobinic acid. The HCO3 will diffuse out of the RBC into the plasma because then it can flow freely to the lungs. And in return, Cl minus is exchanged for it to keep the charges balanced. And that's your chloride shift. When blood gets to the lungs, the HCO3 minus and that H plus recombine. You can get H2CO3 and then it becomes CO2 and H2O because we want to get it to CO2 so we can breathe it out. So here then, if we have a look, hydrogen's not got a negative charge. Hydrogen carbonate does, and chloride does. Hydrogen can bind to the hemoglobin. Hydrogen carbonate doesn't. Remember, that's moved out into the plasma, and chloride it doesn't bind to hemoglobin either. It just moves into the red blood cell. And then it's the product of the dissociation of carbonic acid. So hydrogen protons are. I don't really. It says I on there, so it's not just hydrogen in the element. Hydrogen carbonate is the other one. And then involved in the chloride shift, well, HCO3 is pumped out into the plasma in return for a Cl. So there you go. That's how you do that. I hope that helps. Thank you very much. Please like, share, and subscribe. You know the drill.